So today we're going to talk about heredity, or how traits are inherited. And we're going to try to link this up to our understanding of meiosis. So some basic principles of heredity are that traits get inherited from parents, that each parent passes down one copy or one allele, one version of a gene, with the exception of sex-linked traits, which are a special case that we'll talk about at the end of this video. And third, that some genes or some alleles, some versions of genes, are going to be dominant over others, meaning that when that gene or that allele is present, that version of a trait will be expressed. So normal cells, normal autosomal cells, cells in all the parts of your body except for sex cells, have pairs of homologous chromosomes. So we have chromosomes 1 through 23 in humans. For every single one of those chromosomes, you have two copies. And those two copies, those two homologous chromosomes, are slightly different. So they have slightly different versions of genes. And these different versions we call alleles. So here in the image, for example, the yellow and turquoise genes are at a specific location on the chromosome. That's what a gene locus is. But they have slightly different versions of the protein that they're going to make. So they have different alleles. By contrast, sex cells, so sperm and egg cells, are haploid. Our gametes are haploid, meaning that instead of having homologous chromosomes, they have only one chromosome for each of those chromosomes, 1 through 23. And we saw how that happens during meiosis. We saw that at the end of meiosis 1, our homologous chromosomes separate, so the resulting cells have only half the amount of information as the original cell did. And this is going to affect how traits get passed down. Want to look a little closer at meiosis one more time. So this shows us the basic flow of chromosomes during meiosis, not all the different phases, but just tracking out which chromosomes we'll see. So here it starts with interphase before meiosis. We have two chromosomes, a homologous pair. They're in this sort of loose chromatin format. They're not condensed into chromosomes yet. But they will condense. They'll get copied during S phase of interphase, so they'll undergo DNA replication. Then we'll see that there are two chromatids in every one of those chromosomes. So we see this here in meiosis 1. So we have two chromosomes still, but every chromosome has a double copy of every gene. So this right leg and left leg of the red chromosome are exactly the same as each other. Same thing with the blue chromosome. Red and blue here are a homologous pair. Meiosis 1, they separate. So like this red chromosome goes to one cell, the blue chromosome goes to the other cell. They still have sister chromatids within them, so they still have two copies of whatever's on that chromosome, but they now lack the other allele, the other version of that gene, because it's in this second daughter cell. And then in meiosis 2, those sister chromatids divide. So one copy from that daughter cell from the end of meiosis 1 ends up in this cell. The other ends up in this cell. And same thing for daughter cells 3 and 4 here. They each get one of the sister chromatids from that blue chromosome. So our end result is that each of these 4 daughter cells at the end of meiosis 2 essentially has one chromosome and one chromatid from each homologous pair. So they have one version of every gene. They have one allele, in contrast to the original cell, which had two versions, one on the red chromosome, one on the blue chromosome. This is important because during sexual reproduction, two gametes come together in a process we call fertilization. So an egg cell, which is a gamete, gets fertilized by a sperm cell, which is another gamete. And when they combine, they form something called a zygote. So zygote is just that first cell that has information from both the two parents. Okay, so now that we have one chromosome coming from one gamete, and we saw on the previous slide how you end up with only one chromosome in each daughter cell, and a second chromosome coming from the other parent, we now have two homologous chromosomes in that zygote again. So that zygote is diploid. What trait that zygote is going to express is going to be dependent upon probability. And I'm pretty confident that you've seen Punnett squares before. We'll get to them at the end of this video, or relatively soon actually. But I wanted to show you how Punnett squares are related to what's going on with the cells. So what I've done here is on the left, we've drawn out the two versions of the chromosome that the cell could pass down, the female parent, let's say. I'm gonna flip back to the previous slide for a second. So we see that although there are 4 cells at the end of meiosis 2, 2 of them are copies of each other. So we have 2 gametes of these 4, or 50% of the gametes, have this red allele. 50% of the gametes have this blue allele. So when we do a Punnett square, we don't have to draw out 4 cells because 2 of them are copies of each other. So here we have this red chromosome, these red alleles, this blue chromosome, these blue alleles from the female. The same thing from the male, the exact same process. Meiosis is the same in males. I've made these chromosomes a little darker so that we can see them and track which ones are coming from the male parent in this Punnett square. So we can see that all we're doing is making fake zygotes, right? So if our zygote gets this red chromosome from the mother and from the father, has 2 red chromosomes, right? And then we just go across. So another option is that it could get a red chromosome from the mother and a blue chromosome for the father. All right, so that's the square. So that's that zygote, has 2 different versions of the gene. And then we just continue that process on the bottom part, right? Well, what if you got a blue chromosome from your mom and a red chromosome from your dad? Or a blue chromosome from both your mom and your dad? That's what we're doing when we're making a Punnett square. It's just usually we're looking at a specific trait, but what we're really tracking is just all the possible zygotes that could be formed when those 2 gametes come together. Okay. So what trait or gene someone inherits is going to be dependent upon probability, because all a Punnett square is showing you all the possible choices. And we can see that sometimes if you inherit a blue chromosome from your mom and a red chromosome from your dad, that's not really different than inheriting a red chromosome from your mom and a blue chromosome from your dad. In both cases, you end up with 1 blue and 1 red. So ending up with 1 blue and 1 red, just by probability, is going to be more likely, because out of these 4 possible zygotes, 2 possibilities have this red-blue combination. Okay. And we'll look at that a little more as we track specific genes. Another thing to note before we get to those specific genes is that when a cell has 2 copies of a gene, so it has 2 different alleles, so say we have the red gene and the blue gene, one of them is often, I'm going to even say usually, going to be dominant over the other. So dominant genes, if they're present in that zygote, if they're present in the cell, that's the version you're going to see. So for a lot of traits, right, you can't have like blue eyes and brown eyes, right? You either have blue eyes or you have brown eyes. You just see one color. So what I mean when I say something is dominant, I mean that that's what you're going to see, even though you may have a different version of the gene, a different allele kind of hidden underneath. So that version that's hidden would be called your recessive gene or your recessive allele.